Good afternoon, everyone. We'll be starting with the lecture, and in the last lecture, we have done about the uh, RNA decay and post transcriptional regulation of the RNA. Okay, so now we'll be continuing with that. So, we have left with the epigenetics wala part. So, epigenetics basically like uh, So, epigenetics refers to the stable changes in the cell function that can be inherited. Okay, so without changing any sequence in the DNA. So, this is the basic definition of epigenetic changes. And what happens? Epigenetics basically depends upon the environment. Okay, without changing the sequence of the DNA. So, all the changes that are due to the environment in the expression pattern of a protein without changing the sequence of the DNA refers to the epigenetic changes. So, they can be during the development due to any environmental chemicals or drugs or aging or diet. Okay, diet also plays a major role in the epigenetic changes. So generally what happens, this is the chromosome, and while as we yesterday studied about the um, methylation or the methylation of histones or the DNA. So here the methylation occurs for a particular gene and on the histone tail or the DNA. Okay, so DNA methylation, it is also hereditary, and then histone methylation and certain epigenetic factors that are responsible for the cancer, autoimmune diseases, mental disorder, or diabetes. These are the major complex diseases. So complex diseases include both the things, genetic factors as well as the environmental factors. And epigenetic changes involve all the things. The sorry. So epigenetic involves both the things, the histone modification, DNA modification, use of LNC or microRNAs for gene regulation. These are the stuff that we studied yesterday are involved in epigenetics. So now we'll be moving on one concept, that is X inactivation. So this is due to the formation of an LNC RNA. Okay. So what happens? Why this X inactivation happens? Because we have two X chromosome in females. So as you know, the human female chromosome count is 44 plus XX, and male is the 44 plus XY. Okay, so these are the sex chromosome, and these are the autosomes. So here there are two copies of the X chromosome, and if both the copies express itself, then these lead to the production of proteins that are twice the number of gene products. Okay, so this twice expression leads to embryonic lethality. So to equalize the expression, the nature have evolved a mechanism so that one of these X chromosome is like repressed to express of its gene. Okay, this is also called as dosage compensation. So here is the explanation of the um, in case of mammals, we have only one X that expresses itself, but the other X does not express itself. While in case of Drosophila, both the X chromosome express itself, and same is the case with the CLENs. Okay, so this is the case with the human vertebrates only. Okay, so now how this X inactivation happens. So there is an example of the cat. Okay, so here what happens when during the early embryo there will be a pair of X chromosome, one allele with have which have the orange fur and one allele which have the brown fur. Oh, sorry. So here, what happens when this cell division occurs and the gametes are formed? One would have the orange fur and one would have the black fur, and the other one is the inactive chromosome. So which one of the chromosome becomes inactive? For example, here the orange fur one becomes inactive and here the black fur one becomes inactive. So here whichever becomes inactive leads to the expression of the other allele. So if black ones become inactive, then the orange one will be expressed, and if the orange one becomes inactive, then the black one will be expressed. So here this leads to the formation of a chimeric kind of structure. As you can see here, it has the orange colored cell and here it has the black colored cell. Okay, so this leads to the chimeric structure, and here we have different kinds of cell depending upon which type of chromosome is being inactivated. Okay, so this was the case with the cat. Now, how this thing happens? So, this an X inactivation is due to a long RNA, that is X-ist. Okay, so this X-ist is the RNA that leads to the covering of the X chromosome and it inactivates the X chromosome. Okay, it is around 23 kb in mouse and 35 kb in the humans. It is both spliced and polyadenylated, hence it is produced by the RNA polymerase 2, and but it does not code for any protein, so it doesn't have any ORN. Okay, sequence conservation is not that great between mouse and humans. It's 49% only, and it is associated with the inactive X chromosome. And this RNA is being produced by both the chromosomes. Okay, so one RNA, one X chromosome produces exist RNA and the other also produces exist RNA. But either of them is one of them is to be inactivated and the other is active. So this particular RNA is being stopped by the antisense, its antisense transcript called as T6. Okay, so this binds to this particular RNA and leads to its degradation. But this binds to the X chromosome and it cover itself, so that it will not be like uh, it will not be get activated and expressed in the female body. Now, how this is decided that which X chromosome is to be inactivated. So what happens there, as we have discussed before also, there are certain hypermethylation regions. So which of the chromosomes have this hypermethylation region will prevent the formation of T6 RNA, so that this T6 will not be able to bind to exist and leads to its degradation. So this will be active and this can prevent the X chromosome from expression. Okay. So as we can see is this. So both the chromosomes produces T6, sorry, exist, and either one of them is stabilized, and this leads to the covering of the X chromosome, and here it is the inactive X, and the other one, the excess RNA, leads to its degradation because of the T6 RNA, and this chromosome express itself. So this was the case with the X inactivation. Now this leads to the part end of the transcription wala part. Okay. Now we'll be moving on to the next point, that is nuclear transport. So when now where are we? So this is the nucleus. Here we have the DNA, double stranded. Here the RNA is produced, and this RNA is processed with capping and splicing as well as tailing. Okay, now this has to move out to the cytoplasm for protein synthesis. So now what happens? The nuclear membrane has a pore called as the nuclear pore, and this is associated with many proteins. So this is called as nuclear pore complex. Okay, so these are the proteins. It is the cytoplasmic side, and this is the nucleoplasmic side. And then we have the this is a double layered structure, that is the nuclear envelope. This is the outer and the inner nuclear membrane. Now the inner portion of the pore forms a nuclear basket, okay, which have a nucleoplasmic ring and the inner spoke ring that is bound by the central transporter proteins. Okay, and then we have the we have the nucleoplasmic ring, we have the cytoplasmic ring, and the proximal filaments. Okay, so whenever a protein has to be transported, it needs to bind to this central ring, and then central transporter, then it would have certain signals that leads to the identification of these particular repeats and the release into the cytoplasm. The same thing happens with the export import also, where some the nuclear localization signal binds to these particular proteins that recognizes those signal and leads to the entry of those particular proteins only. Okay, so how this the exit and entry of the mRNA takes place? It takes place with the help of a protein called as RAN-GTP. So this is a monomeric GTPs. So it functions in the presence of the GTP. So whenever a cargo needs to be imported inside, the protein is bound to GTP, and whenever the work is done, the GTP is hydrolyzed to GDP. Now here we have the nuclear exit signal. Okay, so the nuclear exit signal is basically the so there is um, like there are some FG repeats. FG means phenylalanine and glycine repeats in the nuclear core complex. So any protein that needs to be exported out of the nucleus need to have those FG repeats. So these FG repeats interact among themselves. That is then only it is being transported outside the nucleus. So here in the nucleoplasm we have the RAN-GTP. Now this RAN-GTP has first RAN-GTP is formed that binds to exportin 1 and the cargo that needs to be exported out. So these three forms a complex that is called as the cargo complex that have a GTP. Now this interacts with the nuclear core complex and transports it to the outside. Okay, now this export, after this export is done to the cytoplasm, now the guanine exchange factor comes. Sorry, this leads to the hydrolysis of GTP to GDP and the release of the cargo on the cytoplasm. And this now this ran GDP and the exportin one is transferred back to the nucleoplasm via the same nuclear pore complex. Okay, so this was the simpler version of how the cargo is exported out to the cytoplasm. Okay, now moving on, this is how the mRNA is exported to the outside. So in case of the mRNA export, what happens, as we have discussed before also, this is the final RNA, and then we have the protein. So we have cap binding protein, we have poly A binding protein, and some proteins that are RNPs, mRNA binding proteins. Okay, so these proteins are bound to the RNA that helps in its export. And this there is another protein that is an mRNA exporter. So this has three domains: N terminal, C terminal, and the middle domain. That basically has the FG repeats. Okay. So these proteins interact with a large protein complex that have the FG repeats that helps in the like binding to the nuclear core complex proteins and export of the RNA to the uh, from the nuclear core to the cytoplasm. Okay, so this is how the mRNA is transported outside the nucleus. Now this uh, RNA is like after exiting the nucleus, it is again bound with the PABPs, cap binding proteins, and mRNPs, releasing the nuclear versions of these proteins, and cytoplasmic version of these protein bounds to the mRNA. Now, further processing, we'll be discussing about the protein synthesis and how protein synthesis takes place after the RNA is transported to the cytoplasm. Okay, so now we'll be starting with the protein synthesis wala part. That is the last topic from this course. So, in case of the central dogma, when we discussed, so we have discussed about the formation of DNA to RNA, that is transcription. Now we'll be moving on from the uh, copying of the information from the mRNA to the proteins. So the process of carrying the information from the messenger RNA to the proteins in the form of amino acids is known as translation. Okay, and this is done in the form of codons. So codons is like till now it has been like used as a triplet is the used as a codon. So this triplet codes for one amino acid. So codons are three base units that codes for one amino acid. So first we'll be discussing about the genetic code. Further, how this genetic code translates the information in the form of amino acids. Okay. So moving on, we have the genetic code that was framed by George Gamow, but many other scientists have the role in deducing the genetic code. So it is a three-letter code, three-letter base code, which codes for 20 amino acids forming the protein sequence. So how this we have 64 codons as of now, and how this codon number has been deduced. So we have four bases. We have 20 proteinogenic amino acid, and if the base is triplet, so how they deduced was if the codon has a single base, one nucleotide, so this will lead to four combination of only amino codons that will lead to amino acids. But this number is so small that it multiple codons will be coded by like one amino acid will be coded by many codons. Okay, that would create more confusion as compared to the now scenario. If we consider the two nucleotide for a codon, so 4 squared, this leads to 16. So the same thing happens because the number of amino acids is 20. So this number cannot fit for the number of amino acids. Now if we consider a codon to be a nucleotide sequence, so this is 64. So now we have sufficient number of codons for like a one amino acid, or an amino acid can be coded by different codons. Okay, so this leads to the uh, the number of amino acids that are basically formed by the sequence of the codons. So this is how they came with the number of 64. Although research is going on that whether a 4 nucleotide or 5 nucleotide codon can also be there, but that is in like research only, not any outcome has come yet. Okay, so out of these 64 codons, we have 61 sense codon that code for an amino acid, and 3 codons are the stop codons. Although there is certain like codon bias and use of the codons in different species, but this is the universal uh, codon that has been used. Codons are unambiguous in nature. That means each codon codes for one amino acid. Although there are various codons that code for more than one amino acid is coded for more than one codon, but each of these codons will code for only one amino acid, not more than one. This is called as unambiguous nature of the codon. Then the sequence of the codon is continuous. For example, if you consider, this is the sequence, for example, is the sequence of the mRNA, and here you have one codon, the second one, the third one, and the fourth one. So this has a continuous nature, and it is not disrupted. For example, if one of the codon is this, then the other codon will be this, so here will there will be a disruption. This is not the case with the codon. So this is continuous in nature. Then the degeneracy. So one amino acid is coded by more than one codon. Okay. So for example, for like we'll discuss, yeah. So for methionine and tryptophan, only one codon is there, but for other amino acids, the number of codons is more. And the non-overlapping nature, the same. So the non-overlapping also correspond to the continuous nature, because this one codon will be AUG, then the other codon will not be UGG. So it will be after this G ends and the other codon is GCG. Okay, so this is the non-overlapping nature of the codons. Moving on to the other properties, that it is a universal codon. That means it is applicable to all the species. Although some differences there, as we'll see in a couple of minutes, but the actual nature is being universal among various species. Then we have codon bias. So codon bias refers to the probability that a given codon will be used to code for an amino acid over a different codon which codes for the same amino acid. So this is species specific. Okay. For example, in as we consider mouse or human, so one of the amino acid is like codon is considered for coding phenylalanine, but in this one, that particular codon is not used, like preferred for the production of phenylalanine as compared to the other codon. Although if that codon will be there, the product phenylalanine will be coded. Okay, it's about the preference between the amino acid, sorry, between the codon. So that is why it is species specific. So this is called as codon bias, and this has been exploited nowadays for the use of various recombinant DNA techniques and the production of various therapeutics. And then for this basis, we have certain codons that code for different amino acids, or there are stop codons. These two are stop codons, but they code for different amino acids that are found in bacteria. So UGA codes for selenocysteine and UAG codes for pyrrolysine. Then this is the example of the variation in the so variation in the codon assignments in mitochondria. So this is the normal cellular code. UGA is a stop codon. AUA is code for isoleucine. AGA and AGG codes for arginine, and CUN codes for leucine. Likewise, CGG codes for arginine. In vertebrates, the stop codon UGA. In some vertebrates, it codes for tryptophan. Okay, AUA codes for methionine, and AGA or AGG is a stop codon. But these two are normal in some of the vertebrates. Okay, same is the case with the Drosophila. In yeast, for example, Saccharomyces cerevisiae, the common brewing yeast, codes UGA for the tryptophan and AUA for the methionine, and CUN, that is for leucine, it codes for threonine. Okay. Now, same is the case with the filamentous fungi, trypanosomes, or higher plants. So there can be species variation also. Okay. Now, this is the major codon table that has been studied, that has been used so far. Okay. So how do we draw this table? So on the left side, this is the first letter codon. So there are three spaces for a codon. The left side is codes for the first letter. The second letter is coded by this horizontal line, and here the third letter comes. We have UCAG, and that is repeat in all the boxes. So here comes the third letter. Okay, due to this third lane, the degeneracy occurs. Okay, now this is how we distribute different codons with the amino acids. So this is the case, and we have three stop codons: UAA, UAG, and UG. And we have only two codons that code for only one amino acid. One is AUG, that is methionine, and the other one is the tryptophan, that is UGG. Okay, the rest we have more than one codon. For example, in asparagine, aspartate, cysteine, glutamate, histidine, lysine, phenylalanine, and tyrosine, we have two codons for each of these amino acids. Then for isoleucine, we have three. For alanine, glycine, proline, threonine, valine, we have four. And for arginine, leucine, and serine, we have six codons. Okay, this is for the degeneracy of the code, genetic code. So degeneracy, as we have studied, one amino acid coded by more than one codon. So this table explains the degeneracy of the genetic code, and this is how we write the genetic code uh, for the different amino acids. Now, moving on, there is one important concept called as wobble hypothesis. So, how this concept came? So, in total, we have 61 sense codons that code for an amino acid. Okay, there are nearly 32 to 35 tRNAs in a cell. In vertebrates, basically, in lower organisms, the number is also small. Okay, so how do we justify the presence of these many tRNAs for these many sense codons? Okay, so what happens? The environment has designed its own ways. So as we can see here, this is the anticodon loop that recognizes the codon on the mRNA. Okay, so this recognition is reverse in direction. So, for example, the mRNA is 5 prime to 3 prime, and the binding is also 5 prime to 3 prime as per the tRNA. Okay, so this is 5 prime and this is 3 prime. Okay, So how is the degeneracy occurred? So degeneracy occurs only at the third base of the codon. Oh, sorry. How wobble hypothesis occurs? Wobble hypothesis occurs on the third base of the codon. Okay. So this means that the first two bases must be too specific for a tRNA to bind to this particular mRNA. The third uh, base pair can be different. Okay, how it can be different, we'll see. So in the normal scenario, when one codon is recognized by the other codon, so I think this would be clear that only the third base is responsible for the wobble hypothesis. Okay, so in the normal scenario, when the tRNA anticodon is matching with the codon, so in this case, these two will be same, and here the C will be binding to the G. So that is complementary to each other. Now we have A and U that also we have complementary to each other. So this is the normal scenario. Now, when two codons are recognized by the same tRNA, so in that case, if U is present on the tRNA, that can recognize either A or G. Okay, and if G is present on the uh, G is present on the anticodon loop, this would like recognize C and U. Okay, so this is the case when two codons are recognized by the same tRNA. Now, how can three codons be recognized? There is a base that is inosine. So, during the production of adenine, we have seen inosine also. So, when inosine is present as an anticodon loop in the tRNA, so it can recognize three bases, that is A, U, and C. So, here it can recognize three codons. Okay, so here as you can see, I can recognize A, I can recognize U, and I can recognize C. So here only one tRNA can recognize three codons. Okay, so this leads to the generation of the wobble hypothesis that states that one of the tRNA that have this particular anticodon can recognize more than one codon, such that the third base can be uh, can be given some liberty to like have minimum complementarity. For the first two, the normal uh, base pairing, that is Watson Crick base pairing, must follow. So this is the case with the wobble hypothesis. Okay, now we'll be starting with the procedure of protein synthesis. So this is the basic structure. We'll be going a bit in detail about the structure. So, starting with the first step is the activation of amino acid. So, the amino acid are loaded with ami- uh, the tRNA are loaded with the amino acid. These tRNA will now recognize the mRNA, and the ribosome will bind to the mRNA. That leads to the part of the initiation, and then the ribosome like after the ribosome has bind to the like uh, mRNA, and the first tRNA is also bound, then the second tRNA comes and the protein synthesis occurs. So here this leads to the formation of the peptide bond and further translocation on the mRNA chain. So this leads to the elongation of the translation process. Now when the stop codon comes, the mRNA is released from the ribosome, releasing the final peptide sequence that is translated from this particular mRNA. So here the termination comes. Further, this protein is folded and then protein folding for a particular function. So we'll see this process in detail, starting with the uh, requirements. So what do we require for the translation? First is the ribosomes. So ribosome is basically rRNA combined with proteins. So This is also a part of the ribozyme wala part. We have also discussed that time. So we'll be going brief in this respect now. So this is the crystal structure of the ribosomes present in prokaryotes, and this is the eukaryotes. So in prokaryotes, we have two subunits 50s and 30s, and in eukaryotes we have two subunits 60s and 40s. Okay. So the bacterial ribosome completely is called as 70s, and this is called as 80s. So 70s has a molecular weight of 2.7 into 10 to the power 6 daltons. Okay, it has two subunits 50s and 30s. 30s has 16s rRNA, that is made up of 1.5 kb and 21 proteins, and the 50s subunit is made up of 5s rRNA and 23s rRNA. So 16s helps in the identification of the mRNA during protein synthesis. 23s is the peptide transferase, helps in the formation of the peptide. The eukaryotic ribosome is 80s with a molecular weight of around 4.2 into 10 to the power 6 daltons. It has two components 60s and 40s. 60s has three rRNAs 53, 28s, and 5.8s. These two are the peptide transferase with 47 proteins. And 40s has 18s rRNA, so that is evolutionary conserved, and this leads to the identification of the species and identification of the mRNA during translation. And it has 33 proteins. Okay, so this S value comes from the sedimentation rate. So this is the Sevenberg unit, and this is due to the sedimentation coefficient that we go get during centrifugation. Okay, so this is the basic thing about the ribosome. Then we have tRNA. So as we also discussed, we have the tRNA ka structure. This is the tertiary structure of the tRNA that have a clover leaf kind of structure. So we have an amino acid binding arm that has a CCA trinucleotide at the end that is added post transcriptionally. Then we have the T-Cyc arm, the DHU arm, and then the anticodon arm. We have the extra arm also that is variable in different tRNAs. Okay, so this was the structure about the tRNA, and D arm and the T-Cyc arm. This helps in the positioning during the translation process, and anticodon recognizes the codon present on the mRNA, and amino acid arm carries the amino acid that needs to be bound to that particular codon. And we have 20 different amino acids that are proteogenic, so these are like form a protein sequence. So the amino acids are divided on the basis of their charge. So we have positively charged amino acid, that is arginine, histidine, and lysine. This is a single letter code, and these are the three letter naming of these amino acids. Okay, so this is the negatively charged amino acid, that is acidic. So aspartic acid and glutamic acid, D and E. Then we have polar uncharged amino acid, that is threonine, or serine, threonine, asparagine, and glutamine. So S, T, N, and Q, followed by the okay, uh, amino acids that have do not have any group, that is glycine. Proline have a imine group, and cysteine and selenocysteine. So cysteine has a SH group, and selenocysteine is selenium instead of the sulfur group. So This is G, P, C, and U, followed by the hydrophobic amino acids, that is alanine, valine, isoleucine, leucine, and methionine. And then we have the hydrophobic amino acids, phenylalanine. Sorry, these would be the aromatic amino acids. Okay, so this is phenylalanine, tyrosine, and tryptophan. So that have a ring with their structure. Okay, so these were the 20 different kinds of amino acids that are proteogenic, and we also have a pyrolysine also that have not mentioned here. So selenocysteine and pyrrolysine have been found in the bacteria only. Yet it has no role not found in the eukaryotic system. And we require magnesium ion also because ATP hydrolysis is the energy is used from ATP, so magnesium ion is also used. Now, starting with the translation process. So the first step is the activation of the amino acid. Now, amino acid is activated or added. The activation means the addition of amino acid to the tRNA. So, amino acid plus tRNA in the presence of ATP forms amino acyl tRNA that gives the energy in the form of pyrophosphate. Okay. Now, in prokaryotes, what happens to recognize the first amino acid that has been added on the growing chain? The first amino acid is generally methionine in all prokaryotes and eukaryotes. But to recognize the first amino acid in prokaryotes, this methionine is formulated. Okay, so this amino acid is formulated. So this So here, what happens? The methionine is first added to the tRNA, and then this methionine is formulated using the formyl tetrahydrofolate, and this formyl group is added to the N terminal of the methionine. That is, it forms the N formyl methionine, and this is the first amino acid that is added to the codon. Okay, and this reaction is catalyzed by the amino acyl tRNA synthase synthetase. So this is like ATP requiring reaction because the like adding of the amino acid to the tRNA is energy driven. So these require ATP. Now there are two classes of amino acid tRNA synthetase that catalyze different addition of different amino acid to the tRNA. Okay, so we have class 1 and class 2. Class 1 catalyze the addition of tRNA of these particular amino acid, and class 2 catalyze the addition of amino acid of this particular amino acid. Okay, now what is the difference? We'll see in the next slide. So as you can see, activation of amino acid, when the amino acid is to be activated on a particular tRNA, first it is like adenine is added to that particular amino acid. Okay, so this phosphoryl group, that is alpha phosphoryl group of the adenine, attacks the carboxyl group of the amino acid. Okay, forming 5 prime amino acyl adenylate. So how this thing happens? So first ATP basically attacks the amino acid. So ATP is bound to the amino acid in the form of 5 prime amino acyl adenylate. Okay, so this is the adenylate group, and here the amino acid is formed. Now Here comes the role of the amino acid tRNA synthetases. So for class 1 synthetases, what happens? This carboxyl group attacks the because the phosphoryl group attracts its electrons, so it is now the electron deficient compound. So it attracts this particular hydroxyl group of the last adenine of the tRNA, that is the 5 prime end of the tRNA. So it attacks this, and then this amino acyl group is transferred to the 2 prime OH of the 3 prime terminal A residue of the tRNA, releasing the AMP. So now AMP is released, and then the amino acid is added to the 2 prime OH of the adenine. Okay, not the 3 prime OH, but the 2 prime OH. Okay, so this was the case with the tRNA synthetase class 1. Now, in the second case, what happens? First, the carboxyl group attacks the 3 prime OH. Okay, the 3 prime OH is now added to the amino acid, and the AMP is released. So this is the basic difference between the activity of class 1 and class 2 in tRNA, amino acid tRNA synthesis. So now, as they recognize 2 prime or 3 prime OH, that means there will be difference in the active site. That is why they recognize either 2 prime OH or 3 prime OH of the same adenine residue present on the tRNAs. Okay, so this was the difference between the two classes of the amino acid tRNA synthetase. Now, this is how now what happens when this 2 prime OH is uh, attached to the amino acid due to change conformational change. This 2 prime OH transferred the amino acid to the 3 prime OH. So now the final product will be 3 prime bound amino acid to the tRNA. Okay, so this is the final tRNA, how it looks like after adding the amino acid. So this is the amino acid chain. It is added to the carboxyl terminal. Okay, so the carboxyl terminal forms the bond with the 3 prime OH, and this is the amino acyl tRNA chain. Okay, this is the terminal adenine at the 3 prime end of the tRNA. So this is the tRNA attached with the amino acid. This is amino acyl tRNA. Okay, so now the tRNA are charged with the amino acid. Now they are ready to start the process of translation. So first the initiation happens. So for the initiation, what do we require? We require a 30S ribosomal subunit, then the mRNA that needs to be coded, then the first initiating tRNA, that is formyl methionine tRNA, three proteins that are initiation factors: IF1, IF2, and IF3, GTP because that helps in the binding, the 50S ribosomal subunit, and the magnesium ion to form the initiation complex. Now, how the for the initiation we require mRNA positioning on the 30th ribosome chain. That's how the initiation happens for translation in prokaryotes. So for this, the mRNA has a sequence called as Shine-Dalgarno sequence that is recognized by the 16S rRNA, 3' end of the 16S rRNA. So this sequence is AGG AGGU. So this sequence is recognized by the 16S rRNA, and here due to which the 30S ribosome subunit lands on the mRNA. Okay, so 30S ribosome subunit the first recognizes the Shine-Dalgarno sequence. due to which after this sequence, so after 8 to 10 nucleotides, this sequence, the first codon is there. That's why this 30th subunit is recognizes this mRNA. Okay, so this is how the mRNA sits on the 30th ribosome, a 30th subunit of the ribosome. Okay, so first comes the 30th subunit ribosome that recognize the Shine-Dalgarno sequence with the 16s rRNA. Then in the ribosome we have three sites. Okay, so for simplicity, we have divided the ribosome for three sites. One is the A site, P site, and E site. So A site is the acceptor site, where the amino acid tRNA sits on the ribosome. Okay, P site is the peptide bond formation site. So whenever uh, amino acid tRNA enters the A site, it will move to the P site, and on P site, the amino acid that was be present will form a peptide bond to the amino acid present on the A site. And then comes the T site, sorry, E site. So once this peptide bond is formed, the chain is transferred to the tRNA present on the A site, and this tRNA present on the P site moves to the E site. So the E site is the exit site, and from here the tRNA that is uncharged now, so it releases the tRNA from E site. So now the tRNA that is present on the A site has the full peptide chain that now translocates to the P site. So this is how the three site helps in the translation process. So first, the 30th subunit recognizes the Shine-Dalgarno sequence and then binds two transcription factors, the IF1 and the IF3. Okay. So first, uh, the 30th subunit binds to the IF1 and IF3, then it binds to the mRNA. Okay. So IF1 basically helps to like during initiation, first they have to set up the mRNA, proper positioning of the mRNA to the ribosome. So IF1 prevents premature binding of the tRNA to the A site because that is the acceptor site for the tRNAs. Then IF IF3, this basically prevents, it binds to in the ribosome in such a case that it prevents the addition of the 50th subunit right now. Okay, so it prevents the addition of the 50th subunit. So now IF2 is a GTP bound protein. So when it bounds, it facilitates the binding of the first amino acid tRNA, that is formyl methionine uh, tRNA Fmet. Okay. So after forming this complex, the IF2 binds to the complex with the amino acid tRNA, that is the first tRNA with the formilated methionine. So okay, which is the start codon. So one thing that was missed is that the start codon in prokaryotes is AUG and GUG. Okay, so AUG generally codes for, is mostly codes for methionine, and GUG codes for valine. But in case of prokaryotes, AUG codes for two things, Fmet and methionine. So Fmet is the first amino acid. So AUG when present on the first as a first codon codes for Fmet, and when it is in the growing chain it codes for methionine. Same case with the GUG, when it is the first codon it codes for Fmet. But when it is present in the coding chain, in the growing chain, it codes for valine. Okay. So now here the first tRNA recognizes the start codon. with the help of the IF2 GTP. Now, the 50s subunit associates with the 30s subunit, and this is due to the hydrolysis of the GTP. So now the GTP is hydrolyzed, that leads to the addition of the 50s subunit to the 30s subunit, and here all the three initiation factors are released. Okay, why IF1 is released? To facilitate the entry of all other tRNAs that needs to be bound to the A site, and IF3 released due to the addition of the 50s subunit. Okay, so this is the functions of all the three initiation factors and how they help in the process of initiation complex. So now this is the 70s initiation complex with the small subunit, large subunit, and the formilated methionine tRNA that is bound to the start code. Okay, now comes the process of the elongation. So elongation is divided into three steps. First is the addition of the other tRNA, then the formation of the peptide bond, and third is the translocation to the E site. Okay, so for the first case, this is the initiation complex. Now comes the other tRNA bound to the other amino acid with the help of an elongation factor, that is TUGTP. So TUGTP helps in the binding of the next T amino acid tRNA that codes for the other amino acid based upon the next codon. So there is how this TUGTP helps in binding of this tRNA amino acid tRNA. So there is a complex called as TSTU. So the TU is GTP bound when it is inactivated. Okay, so when TS binds to this, so when this is GDP bound, it attracts TS, and when it needs to be activated, so TS basically helps in activation of TU. So TU now gets at GTP and TS is released. So now this is TU GTP, and TU GTP helps in binding of the amino acid tRNA, thus hydrolyzing this GTP. Okay, now this GTP hydrolysis leads to the binding of the amino acid tRNA that codes for a next codon. Now, as this amino acid has been bound, now comes the formation of the peptide bond. Okay, so this is the P side, this is the A side. This is the tRNA structure, and this is the amino acid that needs to be there. So this is thermolylated methionine, and remember, the protein chain formation always occur from N to C. Okay, so the first amino acid will have its N terminal free. Okay, so this like we have for RNA and DNA synthesis, the DNA synthesis occurs in 5 prime to 3 prime direction. The protein synthesis occurs in direction from N to C. So here, this is the formilated methionine, and this has the N terminal free. There is a carboxyl terminal that is linked to the 3 prime points of the tRNA. Okay. Now the same thing happens with the other tRNA that is bound to the second amino acid. Now here, the N terminal is a nucleophilic favors a nucleophilic attack on the carboxyl carbon, and this nucleophilic attack leads to the generation of the electron deficient nitrogen, so that's why it acquires a positive charge. So for this particular rearrangement, the peptidyl transferase comes into play. Okay, so this transfer leads to the destabilization of different bonds, and due to which this particular carbon, sorry, this particular bond is like hydrolyzed, and here it is connected to this particular amino acid. So this F-met is shifted to this particular tRNA, forming the peptide bond. So this peptide bond and the rearrangement is catalyzed by the enzyme called as peptidyl transferase, that is 23S-rRNA, present in the 50S subunit. Okay, so now as this is formed, now the tRNA needs to be translocated to the E site, and this tRNA that is present on the A site translocate to the P site. So that is why now you remember that P site leads to the formation of the peptide bond between these amino acids. So now the translocation happens. So this is the tRNA with the growing peptide chain. Now this moves with the translocation factor, that is EFG, that is also GTP bound. So now this EFG binds to, sorry, so this EFG binds to the RNA chain with the hydrolysis of GTP and helps in the movement of the mRNA on the ribosome chain, such that the tRNA present on the A side moves to the P side, and on the tRNA present on the P side moves to the E side, leads to its exit. Okay, so this EFG helps in the translocation of the mRNA chain on the ribosome. So this also requires GTP for its binding and the movement of the ribosome. So this is how the translocation happens on the on the mRNA for the protein synthesis. So this happens till the amino acid chain or the ribosome reaches the stop codon. Okay. Now comes the process of release as well as the termination of the translation. So now as the stop codon reaches, there will be no tRNA for the stop codon. Okay, so this will be empty. So there is another protein called as release factor that have similar kind of structure as there of the tRNA. Okay, so it recognizes the stop codon and binds to the A position when the stop codon is there. Okay, now this release factor entry leads to the release of the peptide chain that have formed till now. Okay, so this link is hydrolyzed, and then the EFG GTP comes, and this leads to the release of the uh, release factor, the tRNA associated with it, and the mRNA, followed by the 30S and the 50S subunit. So here the release factor and the EF elongation factor G plays a role in releasing the peptide chain and all these factors that are bound to the mRNA chain. So this leads to the termination of the translation process. And here the end of the prokaryotic translation occurs. Now we'll be moving on to the translation in eukaryotes. So the process is nearly similar to that of prokaryotes, but there are a number of factors that are involved more in the process. So here what happens, the in case of eukaryotes, we have additional components, like we have the cap, we have the start codon, and there is a sequence that is present on the eukaryotic mRNA, like the Shine-Darganos sequence in the prokaryotes. There is a sequence called as the Kozak sequence. Okay, so this is the Kozak sequence that helps in the binding of the ribosome and the initiation factor to the mRNA. So first, the this is separate with the formation of the protein complex. So first, the protein called as EIF4, sorry, EIF4G, EIF4A. So what is EIF? That is eukaryotic initiation factor. Okay, so this is named EIF4G. EIF4A comes and binds to this particular region between the uh, after binding the protein E. Okay, so now there is a protein called B that releases the sequence, and this is identified by these initiation factors. Okay, so EIFE binds to the 5' cap, and it is a part of the EIF4F complex. Then we have G that binds to EIFE and to PABP. So what happens? This is a circular protein. Okay, so we'll discuss later like this. So G binds to the PABPs, as you can see here. Okay, so this is how it facilitates the translation. Then we have the A. So this binds to the A site of the ribosome. Okay, like IF1. Then comes the B. So B helps in the binding of the 40s subunit. Okay, so simultaneously the small subunit of the ribosome, okay, binds to EIF1, EIF3, EIF5, EIF1A. Then this is the protein complex, followed by the EIF2 GTP helps in the binding of the with amino acyl tRNA. Okay, so like we have the IF2. So here EIF2 helps in the binding of the amino acyl tRNA to the 43S initiation complex. So this is the protein complex that is formed before the mRNA is being recognized by this particular amino acids. Sorry, this particular proteins. Now this proteins for like recognize this complex on the mRNA and binds to the mRNA. Okay, so this leads to the formation of the initiation complex. So what is the role of different initiation factors? So EIF1, it binds to the E site of the 40S subunit, so that EIF2, that is tRNA GTP ternary complex, can be formed. Okay, then EIF1A. So this is EIF1A that binds to the A site. EIF2 is a GTPase that helps in the binding of tRNA. Then EF2B we have already discussed. EF4F is a complex consisting of 4E, 4A, and 4G. So here 4E, 4G, and 4A binds to the uh, mRNA. Then the 4A has RNA helicase activity that removes the secondary structure. So this RNA forms the secondary structure. So it releases the secondary structure, and then EIF4B helps in. So this EIF4B helps in finding the Kozak sequence and the first initiation codon or the start codon for the translation. So this EIF4B helps in finding the start codon. And then we have EIF5 that promotes dissociation of several other initiation factors from 40S or as a so that 60S subunit can bind to form the 80S complex. Okay, then 5B has GTPase homolog of IF2 that promotes dissociation of the initiation factors before final ribosome assembly. Okay, so this is the pre-initiation complex. Now the large subunit will come and bind to this particular complex, forming the initiation complex. That is ATS initiation complex. Now this 43S initiation complex bind to the mRNA, and then comes the large subunit. Okay, and all the translation occurs in the circular way, so that the degradation is minimal in case of this in eukaryotes. Okay, so, and the second case is the G that is bound to the like the E protein. This is the EIF4 gene that has strong association with the polyA binding proteins. Okay, so that is why this circular translation in eukaryotes occurs. Then comes the elongation. So elongation is similar to prokaryotes, only some elongation factors are different in prokaryotes and eukaryotes. But the other processes like formation of the peptide bond and the translocation is similar to that of the prokaryotes that we have discussed previously. Now comes the process of termination. So what happens in termination? We have a So now in termination we have certain proteins. So we have the eukaryotic release factor 1, that is also GTP bound, and this binds to the A site, and then this GTP, there is a so the EIRF1 binds along with the EIRF3. Okay, so that is a GTPase. So it has GTP bound to it. When this binding is complete, the ERF is released. ERF3 is released. This ERF1 has a GGQ site that helps in the release of the peptide chain of the eukaryotes. So this releases the this releases the peptide chain, and now the release factor comes, that is RLI1, that comes, and this helps in the release of all the components, that is ERF1, the tRNA, the 50s and 30s subunit, and the mRNA. Okay, so this RLI1 protein helps in the release of this particular uh, like complex of the mRNA, the subunits, ribosome subunits, and all the proteins associated with it. So this leads to the termination of translation in eukaryotes. Okay, so now moving on, so this leads to the end of the translation process. Now comes the antibiotics that targets the translation process in prokaryotes. So we have tetracycline that basically targets the A site of the 30th subunit. So it inhibits the binding of the amino acid tRNA binding to the A site. Then we have hygromycin B that targets both prokaryotes and eukaryotes, that binds to the near the A site, and it prevents translocation of A site of tRNA to P site. Okay. Then we have paromycin that targets prokaryotic cell, that also binds adjacent to the A site and the anticodon interaction site in the 30th subunit. So this increases the error rate during translation by decreases selectivity of codon anticodon base pairing. Then we have chromoamphenicol that inhibits the 50s peptidyl transferase in the 50s subunit. So peptide bond formation is stopped. Then we have puromycin that also targets the peptidyl transferase in both prokaryotes and eukaryotes. So this is a chain terminator similar to the chloramphenicol. Then we have erythromycin that blocks the peptide exit tunnel of the 50s subunit. So this also blocks the peptide chain by blocking the exit. Okay. Then we have fusidic acid that targets the EFG elongation factor in prokaryote so that the translocation cannot happen. Then we have thiosteptin that inhibit the factor binding center of the 50s subunit. So interferes with the IF2 and EFG binding sites. Then we have chiromycin that targets EFTU that also is an elongation factor. So this also inhibits the elongation of the translation. Then we have ricin and alpha sarcin. These are protein toxins for both prokaryotic and eukaryotic cells. So this chemically modifies the RNA in factor binding center of large ribosome subunit. Okay, so it prevents the activation and translation factor GTPases. Then we have diphtheria toxin that targets the eukaryotic cell. It modifies the EFTU, okay, elongation factor TU that inhibits EFTU function. So elongation is targeted here. Then we have cyclohexamide. So this also targets eukaryotic cell and targets the peptide transferase center in the 60 unit subunit. So the peptide bond formation is inhibited here. Now this is how puromycin structure is. So this has a structure similar to the tRNA subunit that binds to the amino acid. So puromycin binds to the A site instead of a tRNA that carries the amino acid, and it helps it binds to the peptide bond. It initiates the peptide bond formation, and then what happens? The upcoming tRNA with the amino acid will not be able to form a peptide bond with the next particular amino acid. So it halts the translation process by inhibiting the formation of the peptide bond. So this is how puromycin acts on the prokaryotic cell. So this particular thing leads to the end of our course, the RNA biology. If you guys have any doubt, you can ask me on mail or you can comment in the comment section. Okay, and if you guys need any of these, any of the videos or any topics to be revised, then you also can let me know. Okay, so thank you guys for being here till now, and thank you for your support.